Welcome to the video lecture on protein unfolding for Biochemistry Laboratory 2. Let's get started. The teaching and learning goals are to compute the equilibrium constants for protein unfolding, to correlate the equilibrium constants with the delta G0 values, and to interpret delta G0 values to assess the stability of a protein. Current thinking about protein unfolding is that the unfolded state begins to form local secondary structures that then assemble to form tertiary structures. When two or more polypeptide chains come together to give a functional protein, we call that quaternary structure. One way to learn about protein unfolding is to study, one way to learn about protein folding is to study protein unfolding. And we can think about the reaction as going from the native protein state to the denatured protein state. Proteins can be denatured by heat or by chemical denaturants such as urea or guanidinium, as shown here in this slide. We're going to use guanidinium hydrochloride to unfold proteins in next week's experiment. To understand why urea and guanidinium hydrochloride cause proteins to unfold, consider the structure of these molecules. Both have enormous potential to form hydrogen bonds. Since proteins are solvated by water, these chaotropes, urea and guanidinium hydrochloride, will compete with the proteins to hydrogen bond to water. At high enough concentrations, both urea and guanidinium hydrochloride have the ability to dehydrate proteins. And because you can get these two molecules in very high concentrations, they will outcompete the proteins for the binding of water. So it's believed that this lack of hydration on the proteins at high urea or guanidinium hydrochloride concentrations is what induces protein unfolding. Our goal for this study is to quantify the equilibrium expression. So we actually want to compute K unfolding values at a variety of different denaturant concentrations. And then we want to correlate each one of those to a delta G0 value so that we can assess the stability of the protein. So again, we're going to think about the unfolding as going from the native protein state to the denatured protein state. So we can characterize K unfolding as the concentration of denatured protein divided by the concentration of native protein. This is also correlated to the fraction of denatured protein divided by the fraction of native protein. It's very difficult to compute at any given point in the experiment what concentration of protein is denatured or native. So we have to resort to other methods to help us correlate what we can measure to what we want to know. So that's what we're describing here in the next series of slides. How can we re-express this K unfolding expression in terms of variables that we can actually measure? Since we're going to use fluorescence, and you can use many other experiments to do this, but we will use fluorescence to do this. We'll describe all of the details in the slides with respect to the fluorescence signal that we want to measure. And there are 2 points that are important here. One is that the fraction of denatured protein and the fraction of native protein have to sum to 1. So that's important to bear in mind. And the other important point is that fluorescence signals are additive. And this is extremely helpful for us because it helps us to deconvolute the contributions of each species in terms of the overall fluorescence that we measure. So in other words, the fluorescence that we measure at any given state is going to be equal to the fluorescence due to the denatured state proteins times the fraction of proteins in the denatured state plus the fluorescence due to the proteins that are in the native conformation times the fraction of those that are in the native form. So since we have this expression, these two expressions rather, we can do some algebraic tricks to try to simplify this a little bit for us. So let's begin by trying to solve for the fraction of protein in the denatured state. First, we have to rearrange the terms so that we have the fraction of protein in the native state being equal to 1 minus the fraction of protein in the denatured state. So we can substitute that expression in yellow into this equation for F measured. When we do that, we end up with a series of terms that now have F native and F denatured in the in terms of the fluorescence, but only the fraction of denatured is in this expression for F measured now. So you'll see that this new F measured expression has two terms specifically that have the fraction of denatured protein in them. So we want to somehow rearrange this expression so that they are together. When we do that, we end up with an expression that has F, the fluorescence measured minus the fluorescence in the native state, being equal to the fraction of protein in the denatured state times the fluorescence denatured minus the fluorescence native. So when we solve for the fraction of protein in the denatured state, we get an expression that is exclusively in terms of fluorescence measurements, that measured and that native or denatured. And that's what we want, because these are terms that we can actually measure. Now we can use that same logic to solve for an expression for the fraction of protein in the native state. And when we do that, we end up with the mathematical expression shown in the box at the very bottom. The fraction of protein in the native state at any given point in the experiment is going to be equal to the fluorescence that we measure at that point in the experiment minus the fluorescence of the protein in the denatured state divided by the fluorescence of the protein in the native state minus the fluorescence of the protein in the denatured state. So all of the capital F's represent fluorescence terms, the lowercase f's represent fractions. When we put these equations together to try to come up with an expression for K unfolding, which is what we really want to do, we remember that K unfolding is defined by the fraction of denatured proteins divided by the fraction of native proteins. And we can look at these expressions side by side for the fraction of denatured protein and the fraction of native protein. And what we discover is that the denominators are very similar. For the fraction of denatured protein, the denominator has the fluorescence of denatured minus the fluorescence of native, whereas the fraction of the native protein has the reverse expression in its denominator. So we can easily flip-flop those expressions around so that we have the fluorescence of the native minus the fluorescence of the denatured being converted to the fluorescence of the denatured minus the fluorescence of the native. So in other words, we're going to keep the ratio for F sub D the same, the fraction of protein in the denatured state the same, but because we're multiplying when you divide a number, it's like multiplying by the inverse of it. So now the blue term on the right is in the numerator, but we're going to multiply by -1 over -1, which doesn't change anything mathematically, but it reverses the terms so that we end up with the fluorescence of protein in the denatured state minus the fluorescence of the protein in the native state in both the numerator and the denominator. Those are shadowed in yellow, and they can then cancel out. So now we get a simplified expression for K unfolding that's in terms of different types of fluorescence measurements that we can have. The fluorescence that we measure at any given point minus the fluorescence in the native state divided by the fluorescence in the denatured state minus the fluorescence in the measured state. So this is now a way to describe K unfolding that you can actually compute the value for. So how do we then calculate K unfolding? In order to do that, we have to actually know the fluorescence in the native state and the fluorescence in the denatured state. And we get that directly from the data. So let's pretend that this hand-drawn graph is what your data looks like. It would be a plot of the fluorescence as a function of your denaturant concentration. And let's say you get this curve that's shaped like this, where it begins high, there's an inflection point, and then it tapers off. At no denaturant concentration, you're going to assume that that is the fluorescence where the protein is totally in the native state. When the fluorescence reaches its lowest point, it's going to taper off and never change it at that point. It's going to flatline. And that will occur when the denaturant concentration is at its highest state, let's say in this example. So that means that at the highest denaturant concentration, that's when you're going to have the fluorescence of the denatured state. And so you can actually use your data from this graph to figure out the actual values of the fluorescence of the native state and the fluorescence of the denatured state. Now, one caveat I should point out is that not all data is going to be perfect. So sometimes your data can actually be noisy in these extreme regions. So for simplicity, just take whatever denaturant concentration gives you the very highest and the very lowest, the absolute highest and the absolute lowest values as your F native and F denatured extremes. If your curve happens to look more like an S-shaped curve and is essentially the mirror image of what we have here, you will just reverse these terms so that logically they fit your data. And I can explain that more when we get into class next week to hopefully make that a little bit clearer. Okay, so now you know how to compute K unfolding. And what you'll essentially have, let's say you have 20 different urea concentrations ranging from 0 to 6 molar. Then 2 of those values are going to be considered the values you use to compute the extremes, the fluorescence at the totally native state and the fluorescence at the totally denatured state, which means that you will only be able to compute K unfolding for 18 of those values. So you'll have 18 different K unfolding values, let's say. And for every one of those K unfolding values, you can compute a delta G unfolding. And the way you can make that calculation is to recall that delta G unfolding is minus RT natural log K. And the terms for delta G unfolding are described here. Delta G0 is your standard Gibbs free energy of the reaction. R is your gas constant. T is the temperature in Kelvin. And then K, of course, is the equilibrium constant for unfolding that you've just calculated. This is assuming pressure is at 1 atmosphere. So you will have a series of K unfolding terms and a corresponding series of delta G0 terms based on the data that you generate. Ultimately, your question is, how stable is my protein? And that's really the essential question of the whole study. You want to be able to describe the degree to which your protein is stable. And so you'll recall that delta G0 unfolding is going to be spontaneous when the value of delta G0 is negative. So when the value of delta G0 unfolding becomes less than 0, you describe that as spontaneous unfolding at that point. And so what you want to do is to consider at what denaturant concentration does delta G0 unfolding become spontaneous. So if we looked at some mock data, and I just um, drew some graphs here just to illustrate this point. If you graph delta G0 as a function of your guanidinium hydrochloride concentration in molarity, with 6 molar being the highest limit, and because you get your data is going to have a negative slope, so your data is going to look like the dotted line in this graph. Wherever that line crosses the x-axis is where delta G0 starts to become negative. And so what you want to do is think about what is the guanidinium hydrochloride concentration at the point where delta G0 becomes negative. That's describing to you how much denaturant you have to add in order for this protein to completely unfold. So you're going to get spontaneous unfolding at a very low concentration of guanidinium hydrochloride in the graph on the left, whereas the graph on the right depicts spontaneous unfolding at a much higher guanidinium hydrochloride concentration relative to that on the left. So you're going to use this logic to help you come to your conclusion. The higher the denaturant concentration at which delta G0 unfolding becomes spontaneous, the more stable your protein is. And you can think about this in absolute terms or in relative terms. A urea or a guanidinium hydrochloride concentration that's up to, say, 8 molar is the highest that it can possibly go. Since you're using a stock concentration that's 8 molar and diluting it, your highest guanidinium concentration is probably going to be around 6 molar. So if 6 molar is an extraordinarily high concentration of denaturant, and it takes a very high concentration relative to 6 molar in order for the delta G unfolding to become spontaneous, then you're going to say from an absolute standpoint that your protein is very stable. Let's say that the guanidinium concentration is around 5 molar when the delta G value becomes 0. If the concentration of urea has to be that high in order to unfold your protein, you can say on absolute terms that your protein is very stable. Whereas if it takes a much lower concentration, say 1 molar, 2 molar urea or guanidinium to make your protein start to spontaneously unfold, then you would conclude that your protein is unstable in absolute terms. And if it's somewhere in between, you might describe it as being moderately stable. You can also describe stability in relative terms. Let's say, for example, you do your experiment next week, and then you take that same sample of protein and do an experiment after another week, and you look for changes. If there are changes in time, you can start to describe the stability changing in time. If you subject your protein to some kind of unusual condition, you can then describe the stability as a function of that condition, whether it be temperature, pH, or any other kind of control variable that you want to subject your protein to. And for many biologics that are used in the pharmaceutical industry, this kind of experiment is exactly the kind of experiment that would be done in order to determine shelf life, for example, for a protein that is actually a medicine. So this is a very useful type of study that will help you to understand and characterize a protein stability. All right, now I want to wrap up with some final caveats about the nuances of this study. In terms of the experimental design, because we're going to do this using fluorescence, you always have to ask yourself, what fluorophore will you exploit to conduct the study? You can always resort to one of your traditional amino acids that's a fluorophore, whether it be phenylalanine, tyrosine, or tryptophan. But since we're going to study myoglobin, myoglobin has heme in it as a cofactor, and heme actually acts as a fluorophore. So you could choose to exploit the inherent fluorescence of heme to do your study as well. Whatever fluorophore you use, you have to determine its fluorescence excitation wavelength and its emission peak, and decide over what emission range you're going to monitor your experiment so that you can deduce what the fluorescence is. So you want to think of all those things as you design the experiment that you're going to do next week. To learn more about protein unfolding, you can look at a very classic paper that was published in Methods in Enzymology called Determination and Analysis of Urea and Guanidinium Hydrochloride Denaturation Curves by C.N. Pace. This was published back in 1986 and is one of the benchmark studies describing how to do an experiment like the one you're going to do next week. It goes into much more detail and much more sophisticated data analysis that goes beyond the scope of our class, but it's still an excellent paper to read to learn more about what people do to study protein unfolding. This concludes my video lecture, and I thank you for your attention. And I just want to point out that all the artwork that was presented here was reproduced for educational purposes only, and many of the figures came from the textbook Essential Biochemistry in either the second or third editions that was is published by John Wiley and Sons. Thank you very much.